All right, welcome back. I'm going to give you this one little section from chapter 11, but I'm not going to test you on it, but you should go ahead and watch this. It'll be helpful to put things into context later on. So, what we're going to do is talk about concept 11.1. External signals are converted into responses within the cell. So, here we have a gazelle running, being chased by a cheetah. This is, of course, going to cause the release of adrenaline. Another name for adrenaline is epinephrine. And it's this molecule right here. So, as you can imagine, when you get a release of this, you undergo a lot of physiologic changes. And that allows you to, in this case, run faster, be more alert. Um, but let's talk about how that kind of happens. So, with cell signaling, we have both local and long distance signaling. And this is all centered around the idea that as multicellular organisms, our cells have to work together to do a whole job, to make us do what we do. So, in terms of local signaling, some animal cells communicate by direct contact. So cells are in contact with each other and they can send these signals across to each other. In some cases, you have these things called gap junctions, and these gap junctions might allow particular signals to come through from one cell to another to allow a single signal to affect a whole grouping of cells. In plant cells, we have the plasmodesmata, which allows direct communication between those cells. So that is one level of localized signaling, is through junctions. Sometimes it's just a matter of two cells actually being in contact with one another. So one has molecules on the outside or proteins on the outside, or it could be a combination of proteins and carbohydrates that are recognized by some protein receptor. And when one cell recognizes that, it sends an internal signal to do something. There's also a type of signaling where one cell releases something that can diffuse into the environment around the cell and ultimately make contact with its target cells. This is something that we call paracrine signaling. So in this case, what we do is protein 1, the secreting cells, are producing some type of signal, and it's making it in these secretory vesicles. Ultimately, those vesicles will be released to the outside, releasing those molecules, and those molecules will go and find their target cells and communicate that information. An example of this would be various growth factors tend to work this way. So these are factors that tell your cells that it is okay to grow and divide. It's in the right place, doing the right job. All right, so that's paracrine signaling. Another thing we have is synaptic signaling. This is where we have, this is very closely related to paracrine signaling, but in this case, we have two cells that are kind of intimately contacted. And these signals are diffusible and are released in the same way that we see in paracrine signaling, but they can't really diffuse outwards. They can only affect the cell that it is in tight contact with. Now, this is localized signaling when cells are close together. Well, what happens when we need to do long distance signaling? This is where we get this idea of hormones. Okay, so hormones are these long distance signaling molecules that is some type of chemical or biomolecular molecule that is released out and goes out and achieves some type of effect. Now, when we have a hormone that goes out and impacts a lot of cells and it's distributed by the bloodstream, we'd refer to this as endocrine signaling, or we could also call it hormonal signaling. So here we have a cell that in this case is living in the brain, and it is processing signals from your brain. Your brain tells it to release this signal because something is happening. It releases the signal, it diffuses through the bloodstream and gets distributed to the entire body, where that can then contact and affect its target cell. So that's kind of short distance versus long distance. So let's talk about what happens when that signal actually reaches the cell. Okay, there's three overall steps in this process. There's a reception of the signal, there's a transduction of that signal, and then there's the response. So the reception is when you get the signal being directly detected by a receptor. That receptor is always a protein of some form. Oftentimes that protein is on the or is in the exterior of the cell in the plasma membrane, and is often an integral membrane protein, meaning that it goes all the way through the membrane. It could also be inside of the cell. So there are a few kinds of receptors that are inside the cell that can actually detect signals that can freely diffuse through that cell membrane. So if that hormone is something that is fat soluble, it can just pass right through. So this is step one. When this happens, when you get a receptor binding a signaling molecule, typically it will change shape. Once that happens, you initiate what we call transduction. Okay? This is where that signal is starting to be passed ultimately to some type of final destination. This is often a multi-step process. The most readily available version of this is ones that involve phosphorylation cascades. So this protein may start to add phosphate groups to target proteins when it detects its signal. So signal binds, this phosphorylates protein 1. Protein 1 is now active and then can phosphorylate protein 2, which is now active and then can phosphorylate protein 3. This is one example. There are other times where we release some type of small molecule. Calcium ions are often used as a signaling molecule. This transduction allows us to both interpret signals from the surface and then somewhat interpret or amplify or dampen those signals depending on how much of this is happening. So this provides a way of either amplifying or diminishing that signal dependent on the circumstances at the surface. Ultimately, that's going to lead to some type of cellular response. Sometimes that is an activation of a new set of genes. Other times it is the activation of particular proteins that are then going to do some type of work. Okay, so we have a whole slew of different responses. Some of them involve the regulation of what genes we are expressing and turning into proteins. So exterior signals are converted into responses within the cell. We can do that on a local level from direct cell-to-cell contact through gap junctions or plasmodesmata, or by directly contacting each other. You could also secrete a signal, which might go semi-short distances to hit target cells or go localized directly to neighboring cells. We can also release signals that pass through our bloodstream and impact our entire body. And once a cell gets that signal, it goes through three steps. You get reception of that signal, transduction of that signal through a series of relay molecules that will ultimately lead to some type of cellular response.